Good day, everybody. Oh, that's a little bit loud. My name is Brooke Biggin, and I will be your lunch narrator today. Woo! Yes, that's my cheering section there. So I'd first like to acknowledge that we are on the unceded Coast Salish territories of the Musqueam, Tsleil-Waututh, and Squamish First Nations. I've also been asked to disclose any conflicts I have, and although I'm usually at the center of a lot of conflict in my life, I don't have any to disclose, besides the fact that, of course, ViiV has provided some support for my participation in this session. I can still remember the first time that I ever saw somebody scale a government building. It was the same day that I saw people laid out across the floor of cathedral aisles. Both of these people were living with HIV. I saw that these people were mobilized, that they were organized, and most of all, that they were living as vital as humans could be. At the age of 23, having just been diagnosed myself, I didn't know anybody living with HIV. And so these images, these films documenting the early days of AIDS activism were my introduction to what HIV was, how it shaped the world, and how it would shape me. I looked at these people like they had superpowers, and I just knew that this was a cause that was urgent, it was a cause that mattered, and therefore I mattered. And that this was a people that were bold, that were powerful, sexy as hell. I'm serious, go back and check the footage. There's some good looking folks. And effective. Now you might ask why I'm here talking to you while you're just trying to eat your sandwich. And that's a good question. And although I'd like to think I was invited for my charm and good looks, the truth is that I, Brooke Biggin, Prairie Regional Manager with the CBRC, founder of the EMHC, am also somebody living with HIV. And so I'd like to acknowledge the organizers of this symposium, ViiV, for inviting me for that reason, because people living with HIV should always be in the room when we are a topic of conversation. And not, oh, there you go. And particularly today when we talk about the evolution of HIV treatment, the topic of today's session. Because the evolution of HIV treatment is inextricably intertwined with the evolution of us as a people. I'm not a scientist. I scored 67 in high school chemistry. So I was surprised my first time at CAHR when I went to the basic science section thinking that, that was more my lane, to find out that there wasn't much very basic about it. Right? So we do have a great set of panelists who will be here today to talk about the science, but I thought I'd share a little bit about the evolution of treatment from the perspective of somebody living with HIV. And given how different we are, because we're not all the same, and my place of relative privilege, I don't want to claim to speak on behalf of all people living with HIV, but would like to share just a little bit about my own history. The impact of those images, those early days, those people with those superpowers, changed the course of my life forever. I got a job with an HIV organization, started a grassroots queer men's health organization in the vein of my heroes at ACT UP, and I even got the pink triangle tattoo on my forearm and silence equals death across my chest. I would show you today, but I'm pretty sure I'm not allowed to take my shirt off on the stage at CAHR. I don't know, it wasn't in the rules. But now in preparation for this session, I've had to reflect a little bit on my life and where it's gone and what my relationship to HIV now is. And although I still work in the sector, I realize that I spend more days completely oblivious to the fact that I'm living with HIV than I am aware of it. I take 2 pills a day. One is for HIV, the other is for hair loss. And neither carries more symbolic weight than the other. And to be honest, at this stage in my life, I'm probably more concerned with going bald. I've got a denty head, I just can't pull it off. Oh, there's a little laugh from the corner. Thank you. Appreciated. And so what does it mean that my life has shifted and that my relationship with HIV is not what I once imagined that it would be or should be? Have I backslidden? Have I lost my path, sold out? I don't know. And these are all questions that I'll have to sort out over the coming months and years. But I did want to share two thoughts today with you that emerged during my reflection. Usually in these settings, people living with HIV are asked to come and speak about all that's left to do, to light a fire under people's butts. And there are amazing organizations of people living with HIV in this country that are doing exactly that. The CPPN, the grassroots movement behind U equals U. And I'd be remiss with the topic of this conversation to not talk about how unconscionable it is that there are people living with HIV in this country today who cannot access life-saving treatment with ease due to the province they live in or their immigration status. And that is something that we all have a responsibility to address, not just because it's a good public health strategy, but because people living with HIV are people and people matter. But today, before I turn it over to the panelists, I don't want to talk about things that are left to do, but rather what we have already achieved. Funny enough, in preparing for this session, I thought about retirement planning. I'm nearing 30, the horror, and realized that I need a break, and if I'm ever going to retire, I got to get my act together. And so, of course, as you know, if you've gone onto one of those online retirement calculators, in addition to asking about your income and how much money you'll need to live, they ask you how long you plan to be retired for, which is essentially a nice way to say, when you gonna die, right? And so just a couple months ago, I went through this exercise, and I said 105, because why not? You see, I've been living with HIV for about 6.5 years, and not once have I ever thought about the potential that I would die young. And I recognize that if we rewound the clock about 30 years ago, that at 29, I could possibly be in the last few years of my life. But I'm not, and that's not a dumb stroke of luck. It's not fate, but it is a very real and tangible result of the years and decades of work of people living with HIV who scaled those government buildings, who laid themselves out across cathedral aisles so that all of us could live. And so, because I don't know if I'll ever be able to stand in front of you all again after this presentation, I'm going to ask if you'll do something with me before I invite our guests up. If you were involved in what I call the early days, I don't know what the early days are, but essentially, if you were involved in the effort to ensure that there was effective and accessible HIV treatment, do you mind just raising your hand from where you are, whether you are a healthcare provider, you are a researcher, you were a person living with HIV? Thank you. And now, because I see you, if you don't mind, would you mind just standing for one moment if you can? Just one moment. It'll be 10 seconds, I promise. There you go. There you go. Thank you. So, I know that there is a lot of work left to do to ensure that all people living with HIV can experience the benefits of the evolution of HIV treatment. And I know that we are not truly free until all of us are free. But when it's, ooh, I wasn't expecting this. When it's you who gets to live and not die, that matters. So, I want to thank you. And on that note, woo, girl, it's getting hot in here. And so I really appreciate all of your efforts and bringing us to where we are. And so now I'd like to introduce our panelists who will be looking at the following questions. Until we find a cure, how do we progress antiretroviral therapy? Should we try to further improve triple therapy or expand 2-drug regimens? Have we reached a fork in the road regarding HIV treatment? Today we have with us Dr. Jean-Pierre Routy, not Roti, Dr. Jonathan Angel, and Dr. Sharon Walmsley. So I'd like to invite Dr. Routy to the podium.
Brooke, thank you very much, Brooke, and it's a pleasure to be here and to start with the bad part, the beginning of the epidemics. So that's the conflict of interest. So you can see on this slide that it was a dark age, and everything was dark, and it was a very bad time. However, you remember that medication start only in 1987 to the commercialization of the first drugs, zidovudine or AZT, followed 4 years after by DDI, then DDC. So you can see that only in 1995, drugs that we still use, lamivudine or 3TC, was available commercially. This drug or this class of action were blocking the NRTI and blocking the maintenance of into the virus, and a chain termination was blocking the virus at one site. And these first medication were focused of attacking the virus only at one enzymatic site, making the concept relatively weak to attack at the same site. So the first medication were the most important, of course, the zidovudine, follow few years after with the 3TC or lamivudine. They both belong to the family that was called to be NUCs, to be faster than nucleoside reverse transcriptase inhibitors, and they paved the way for the first significant clinical outcome. Remember that the birthplace of 3TC was Montreal and McGill, and in these rooms that you can see, that where in fact 3TC was engineered. And it takes development with a drug company to make this drug, who is the most sold drug for HIV worldwide, and never took a wrinkle since the beginning of his discovery, is still used and still part of standard therapy. And of course, it will be very important to recognize the contribution of Bernard Béliveau, the McGill scientist, biologist who designed the compound, transformed for commercialization by Francesco Bellini, was a very handsome and very, speaking easy, very strength in putting things together. And of course, Mark Weinberg, who discovered with Hugo Swedens, who is seated at the back of the room, the contribution to the resistance with the famous 184V, and all the works have been also studied by Bluma Brenner from his lab. So the first therapy that mattered in history of HIV was in fact the combination of these two drugs doing the first duet. And the duet was studied in France, led by Christine Catlamar at Pitié-Salpêtrière in Paris, studying the safety and efficacy of 3TC and zidovudine combination therapy as in antiretroviral naive patients. So the concept of treating with two drugs and not one was not an easy one, and it was a controversy to use either sequentially the medication or at the same time. And the conclusion was that the combination of lamivudine and AZT result in a potent and sustained, at the time, of course, antiviral effect in never treated patient and better than one drug. And if you go on the website, you can see images are very bad. Everything is black and white, so the technology was not the same. No internet at this time, and showing the different population. What is relevant, you can see on the higher, the B therapy, and the first assessment of efficacy was not viral load, but CD4 recovery, and that was the only marker at that time, or the initial trial, was to assess first the tolerance of the medication and the ability of CD4 cells to augment and to plateau before going down by what is known to be HIV resistance. And when you give double therapy, you can see an increase of 80 cells versus AZT of only 20. And when you fail AZT and give 3TC after, you can see that the benefit is less. So one followed by one improve less the CD4 than combination therapy. So the concept of B therapy was born and confirmed by this large trial. As a second figure follows the CD4 was the viral load. Today we will show first viral load activity because it's antiviral, and then the effect on CD4 recovery. At that time, CD4 was the marker of a good improvement, and viral load was a secondary biological marker. And you can see that one drug dropped to 0.6 log versus B therapy to 1.4 log, showing the benefits of combination on a viral perspective marker, translating by a better CD4 recovery. So that's the same thing on a different, so the delta change of viral load with one or two medications showing the benefits, and also the difference of the slide. So of course, treatment came with some failure, and you can see that failure occur after 6 to 12 weeks. You start to see a mutation, and the famous M184V was present, in a relatively significant proportion of patients, and may have also an effect in decreasing fitness, but that was not identified at that time. So the number of subjects with AIDS, because clinical endpoint was also a marker, and when we see this data, we are shivering how bad, we still have to see if severe side complication of HIV were limited by triple therapy, and yes, there was a benefits on clinical. So then effect on hepatitis B came also at a good time. The research in one domain of medicine, being HIV, can also be a fertile soil for other viral infection, like hepatitis B with 3TC, and shows the importance of the trans field or collaboration. And when something works in medicine, generally, it may work in other conditions. So then, when you look at control trial as a meta-analysis, you can see a significant decrease in viral load, showing the benefits of the combination therapy. So together were different, but still they were attacking the same enzyme, the same enzymatic pathway for the virus, and definitively we need to evolve, and time will tell. And just after this dark age starts a ray of hope, and with having all these molecules since improve, and the very beginning of this improvement occur in this very important city, Vancouver, in 1996, when at the convention center next door, one word, one hope, and that's the best way to give now the talk to Jonathan to put positive things ahead, and that was a change in the way to fight HIV.
Thanks, Jean-Pierre. I don't think you were saying I was the one hope. That was the one hope for at the time. But, thanks very much. And I just want to thank our ViiV for inviting me to participate in this. When I was asked to speak on I'll focus on 20 years of triple drug therapy. I think that's the name of the talk. I was a little curious on why I was asked to do this. This is a bit of a history lesson, I guess, and I wasn't sure that the audience would be that particularly interested in this, but they asked nicely, and so I said yes. So it also gave me an opportunity to look back and reflect on what we're doing today. So when it comes to management of people today, we don't often think about how we got to where we are. We have a lot of options, a lot of will be future options, and the focus on is treatment today. But I think it's perhaps important for this discussion and to lead into Sharon's talk a bit of some perspective of why we are where we are today. And so that's the title of the talk: 20 years of triple drug therapy. And my graphics were like the graphics from that paper, Jean-Pierre. They're white background and black text, and it'll stay like that. There, I've got some conflicts. So I was also asked, how I could do this. I wanted to choose a landmark paper and focus on that and use that to direct the talk. And looking back and talking to others, and I started around the time of when these studies were being done and the studies were coming out. So I was familiar with all this, but not. I hadn't thought about it in a while, and talking to colleagues, and particularly Bill Cameron, who's very involved in the early development of trials and very familiar with how pharma and the investigators and the licensing processes are working. There's a lot of interesting history. I'm not going to go into those details, and I think you could argue which is the first drug or the best initial drug or the best approach. But there is some interesting history, but I just focused on this one. This paper, it's really a paper of indinavir and 2 nucleoside analogs. It's not the first study of 3-drug therapy, but it's the first drug that demonstrated both within the same study, a decrease in viral load and associated with a decrease in morbidity and mortality. So it was I thought it was kind of landmark in that respect. And this, I think you can see this, I just put up the information here. First, it's what's interesting in these studies that were done, and they were done relatively quickly. It's a lot of patients, and that also let me reflect on this. At this time, getting patients enrolled in these clinical trials was a way to get access to drugs, and I think at that time enrollment in clinical trials was life-saving. Now we are involved in many clinical trials. We may be able to fine-tune therapies, provide therapies perhaps with fewer long-term toxicities, perhaps provide therapies that are more convenient to take. But at that time, enrolling patients in trials was really, I think, was related to or resulted in saving lives. The next, so this is just very simple data. I'll go through these couple of slides. This is the viral load data, and to Jean-Pierre's point, today it's standard to do viral load monitoring many times with all these patients. It wasn't standard at that time, and these data show that it's only viral loads on a select group of individuals, but the data are the same, that the triple drug therapy had a greater virologic effect than single drug therapy. All of this we say today, of course, that's the case, but I think it's just important to reflect and see what was done here. And then, what we don't see now, what was done in the past, was really the impact on morbidity and mortality. And this regimen of indinavir and 2 nucleoside analogs decreased AIDS or death and decreased overall death. So that was an important observation in this regard, combined with a number of other observations with other therapies at the same time. And I forgot, I wanted to thank Victor Kramer from Merck for providing me with some of these slides. And then this is just another graph of survival of the same patients. So this led around the time of this and other papers, the guidelines were being developed through the Department of Health and Human Services. There may have been others globally, but this is the American one and kind of the one that are often referred to, the DHS guidelines. This was in 1996, they established a panel to review guidelines for management of people living with HIV. And this is the first statement that came out regarding triple therapy. So I'll just read it. When initiating therapy in the patient who has never been administered antiretroviral therapy, one should begin with a regimen that is expected to reduce viral replication to undetectable levels. And that's the quality of the data. And based on the weight of experience, the preferred regimen to accomplish this consists of 2 nucleoside reverse transcriptase inhibitors and 1 potent protease inhibitor. So that was pretty explicit. And so that was in 98. So then 20 years later, so this is the most recent guidelines. So these guidelines get updated every so often and rewritten every so often. So it's a little bit smaller in the bottom here, but basically it's the same. The guidelines for initial therapy are 2 nucleoside analogs plus a third drug. Initially it was a protease inhibitor, but there's been other drugs developed, obviously. But the recommendations for preferred therapy for 20 years has been 2 nucleoside analogs and a third regimen. So there's been little change in that. And then I'll kind of go over I think is going to go over the reasons why this has been the same. This has really been unchanged for 20 years. So, there's my slide. So why is this approach, the need to start triple therapy, not changed in 20 years? And all this is my opinion about why this is the case. Some of it is factual, but some of this is very obviously my interpretation of why things are as they are today. So firstly, it works, which is, seems quite obvious, but we continue to use triple drug therapy because triple drug therapy works. And it continues to improve. So there's obviously decrease in pill numbers, dosing frequency, and ultimately resulting in single tablet regimens. The current regimens are better tolerated with less toxicity than they were initially, and there are fewer drug-drug interactions with newer therapies. So improvements have been made to the extent that current triple drug therapies are almost perfect. So they're very, very good, but not they're far from perfect, but they're pretty close. And this is a slide that Connie Kim from Gilead provided me, just looking at virologic outcomes with kind of the most current regimens. So this is bictegravir and FTAF compared to dolutegravir and Kyvexa or Triumeq in one arm. And if you look at people that are on therapy, there's about 99% or over 99% of individuals that are on therapy that have suppressed viral load. So that's maybe not perfect, but that's very good. So that's pretty good justification for continuing to treat patients the way we have been with triple therapy because it works extremely well. The next is, and I think this is also important, it's hard to quantitate things like this, but doing this for 2 decades and seeing the results and seeing the improvements, it's reinforced the belief that an effective regimen must have at least 3 drugs and from at least 2 different classes. And for many people, I've just known from hearing them speak or speaking to them, this is a bit of a fixed belief, is that you have to use 3 drugs and that you shouldn't sway from that for whatever reason. It's almost a religious belief. So there's kind of loss of flexibility, I think, or open thinking about what is possible. But the more time has gone on, and the more people have used these regimens that have worked, the belief becomes almost fixed. So I think that's part of the reason it's as it is. So alternatives have never caught on, and there have been a large number of alternative approaches tried for various reasons, some of them obviously very good reasons. So early on, nucleoside or nucleoside sparing regimens, there were individuals and groups that obviously concerned about the toxicity of early nucleoside analogs, and there was a lot of enthusiasm by some, not by all, to study nucleoside sparing regimens, but these had variable degrees of success and variable degrees of buy-in. So they never caught on. There are certainly studies where some nucleoside analogs or nuke-sparing regimens worked, but the whole concept never really caught on. And with newer nucleoside and nucleotide analogs with less toxicity, the enthusiasm for this approach has waned. It still exists to a degree, but it's waned. The next is that big pharma is excellent at marketing drugs, but either are reluctant to develop and can't or won't, and I don't know, develop a therapeutic approach. And I'll give you examples of this. So there's ways that the studies have been done, and many of these have been investigator initiated, but some of these have been pharma initiated studies, where there's approach of giving a lot of drugs up front and then de-intensifying. So you can give 3, 4, 5 drugs initially to suppress viral load and follow that with 1, 2, or 3 drugs. That's an approach, and there are a number of studies looking at that approach, but that approach has never caught on. There are a few studies that look at 5 days on and 2 days off, and I think there's 3 published and 1 presented study, and they've all worked, but that's never caught on. And I don't know, it's because the pharma companies that have these drugs can't market approaches or choose not to market approaches, but it just hasn't. And then twice a day to once a day, where perhaps a drug is licensed or works at twice a day, but if you took it once a day after initial viral suppression, it would work. Again, that's something that hasn't been taken on, but that's the way it is. And I'll give you, this is just an example by one of my heroes, Bill Cameron. One of the papers that was published a while ago was in the era of Kaletra monotherapy. And the study was Kaletra plus 2 nucleosides, and when your viral load was suppressed, you'd stop the nucleoside analogs, versus standard therapy, which is efavirenz-based. And if your viral load rebounded after stopping the nucleoside analogs, you could add them back to hopefully further suppress viral replication. If you look at the results of these, the approach of stopping nucleoside analogs if your viral load was if suppressed and then restarting as necessary was at least as good as a standard therapy of efavirenz. So this approach was at least as good, perhaps somewhat better, depending on what outcome you look at. But this, again, never caught on. Maybe because it's hard to sell or to market, but this is just an approach that works, but just has never caught on. Next, in North America, there's been little motivation for prescribers or pharma to limit drug costs. This is, limiting costs is good on a societal basis, but when it comes to an individual basis, deciding on therapies, I think it's rare that the physician, speaking from experience, will bring in the cost of individual drugs when it comes to the patients. If patients have access to the best or what they consider the best drugs, I think that's going to be an overriding priority when it comes to the individual patients. I think most people think, yes, on a societal basis it's good to save money, but on an individual basis, people would like to prescribe what's best for the individual patients. I think there's other areas in Europe in particular where they're very much more budget driven, that there's a much more of a push to decrease cost or prescribe drugs that cost less. But my experience in North America, and I think it may be even greater degree in the US than in Canada, this is not a big driver. It may be somewhat of a driver, but it's not a big driver to change current practices. And then the last, and again, most importantly, it works. So why would we change something that works? And nothing has shown to be better. There may be other approaches that are as good, but nothing's shown to be better. So with an ever-increasing understanding of HIV pathogenesis, improved technology to measure HIV in the body, and the intense study of many alternative or complementary approaches to treat HIV, there's yet to be anything superior to indefinite treatment with triple therapy. So this, again, this reinforces, I think, our approach to management. And therefore, for a new approach to be widely adopted, it will need to be better than what is currently available. And given the slide I showed you and how effective these drugs are at suppressing viral load, nothing's likely to be better with respect to the degree and durability of viral suppression. So short of a cure, any new approach will have to be at least as good with respect to viral suppression, plus one or more of cheaper, less toxic, or having a preferred dosing formulation, route, or interval. So that's a big, perhaps a big task, but that's where the field is going to have to go if something, I think, is going to replace or be an alternative to triple drug therapy. So it kind of raises the questions: do we need? Sorry, Sharon. It raises the question: do we still need 3 antiretroviral? We still need to use 3 antiretroviral agents. Thanks.
Okay, lunch is almost done. Does it feel a little bit like Scrooge's Christmas Carol? And I guess I'm the ghost of future. So my body isn't living with HIV, but my soul has lived with HIV for the last 30 years. And I must say, having had a career which has spanned this incredible epidemic has been very rewarding. And I just wanted to thank all of the opportunities that I have had, all of the patients that I've had the privilege of working with and caring for and helping me with my research. And further, I couldn't have done any of this work without being conflicted or partnering with all of the antiretroviral companies over the years. So I'm to kind of look to the future and sort of ask the question, are we ready to go back again? And do we still need 3 drugs? So I think John very eloquently told us why we needed 3 drugs. We needed 3 drugs to get that viral load down, and we needed 3 drugs to keep that viral load suppressed. And the main reason for that was to prevent resistance, so that if the virus is growing when there's drug there, that's what leads to resistance, and that's what leads to the failure of the regime and possibly compromises other treatments that you might want to consider. And so we really talk to that as being a barrier to resistance. How hard is it for that virus to become resistant to the drugs in combination? And as John alluded to, for 30 years, our thinking was we needed 3 drugs in order to accomplish that. So why would we go back? And John says it works, let's not break it. If it's fine, let's not change it. Well, if you take less drugs, there's clearly going to be less toxicity. There'll be less in the short term and less in the long term. And the mantra that we're hearing was, well, our new drugs are safer and better tolerated, and sure they are. Absolutely, they're much better than the drugs that we have in the past, but we don't know for the long term, and clearly not taking something is not going to give you toxicity, whereas taking something might. The other reason is if we only use drugs in 2 class, then we've got lots of options. So if something goes wrong along the way and we need to introduce some new agents, there may be some more choices as that happens. Always, why use more than you have to? And as John alluded to, although we have not had the pressures of cost, that's clearly something that we need to think about, and I think we clearly need to think about that as we grow older as a society and the costs of medical care are going to increase. But I think the one biggest argument is we've always thought of barrier to resistance. How hard is it to get resistant by the number of drugs? And it's really not the number of drugs that matter. It's really how hard is it to get resistant to the particular combination. And for some drugs, it's relatively easy, and for other drugs, it takes many, many more mutations. So it's not the number of drugs, but rather that barrier to resistance, which is most important. So it's not that we didn't try, and John explained to you some of the regimens that we tried over the years with 2 drugs, and I've listed them all here, and they all had silly acronyms that we all forget, but they didn't work. And we tried, and it just didn't work, and it didn't make sense. We were putting 2 drugs together, which for all intents and purposes should work, and they didn't. Well, it's not that they didn't work, but they weren't just quite as good as the 3 drug combinations for the population. Sometimes it might be fine for an individual. And probably the one that everyone was sure was going to show us that answer was something called NEAT. And this was a study that was done in Europe. And what it did is it compared a protease inhibitor called darunavir, which was boosted with ritonavir. And that drug was either given with Truvada, which contains FTC and tenofovir, so 3 drugs, or it was given with an integrase inhibitor called raltegravir. And everyone at the time thought, well, darunavir is a real strong drug, raltegravir is a real strong drug, why won't this work? Well, it did. And it did. And if you look at the overall result of the study, the two drugs were what we use in scientific terms were non-inferior. One was not any worse than the other. But when you look at the patients that might have the biggest challenges to antiretroviral drugs, those with the lowest T counts and those with the highest viral loads, so when you really stress something, then we did start to see a difference, and the two drugs didn't work as well as the three drugs. And everybody was sort of ready to give up and go back to the three drugs are what is necessary. But then we started to see some differences. And so Pedro Kahn, who's from Argentina, was really one of the people that started to push this concept of two drugs again. And he designed something called the Gardel study, which was largely a study that was done in South America. And it compared a protease inhibitor called Kaletra or lopinavir, which many of you will know, and combined it either with 3TC alone, the good Mark Wainberg drug, or two reverse transcriptase inhibitors. And what he was able to show is that the people on two drugs were more likely to suppress their virus than people on three drugs. People didn't believe him. They said, you just, you're in Argentina, you use toxic drugs, you didn't really stress it. Well, what about the people, the tough ones? What about the people with the high viral load at baseline? Well, they did even better on two drugs than the people on three drugs. And so the question is why, and is this some way being driven by toxicity because of the extra drug, or is it really that the two drugs were doing just fine? But the world didn't embrace this. Despite showing this in a randomized controlled trial, there were lots of arguments, and the main argument being that we had moved on and nobody was using Kaletra very much, and therefore we couldn't extrapolate these results to what was happening in the field at the time. So Pedro doesn't give up, and so dolutegravir, which is one of the new integrase inhibitors, came on the market, and dolutegravir proved to have a very high barrier to resistance, and he says, okay, I'm going to try it again. So this time, he combined dolutegravir with 3TC, just two drugs together, but this time, he didn't have a comparator arm. He just looked at a small number of people. And in order to be safe and to ensure that we were not compromising their health, he studied only 10 people at the beginning, and when the 10 did fine, then he got 20. But he was scared, and he measured these people to death, and these poor volunteers for this study were being bled every 2 or 3 days in order to make sure that all is well. But what you see in the bright yellow there is how quickly these people suppressed their viral load. And you can see that everyone in the study had dropped their viral load to the undetectable level within 4 to 6 weeks, and it maintained there for 24 weeks, and he's now studied these people out to 96 weeks, and they've continued to maintain their control. Well, people don't believe Pedro all the time, and of course, in the United States, there's a clinical trials group called the ACTG, the AIDS Clinical Trials Group, and they always like to challenge things. And so this was a study that was organized by them to look at this, but now we're less cautious than Pedro and said, okay, let's try this in the people who have higher viral loads and maybe lower T counts and really stress this regimen and see if it's going to work. So he recently has published his study in one of our journals. He has studied 165 people, so I caution you again that it's still small numbers. And this is just the study chart showing how many people started on this drug. And there was 122 people who started study, and it included 37 people with a higher viral load greater than 100,000 and 83 people with a viral load under 100. So again, stressing it a bit, but not stressing it all the way. So caution to the wind. In his small study, 5 of the 120, or 4% of the people, did not have what we would call virologic suppress at the 24-week period. And unfortunately, when we talk about clinical trials, we talk about virologic non-success, we talk about virologic failure, and those terms get confused. But basically, his 2 patients with virologic non-success had started with a very high viral load and were deemed not successful because they had a blip of their virus between 50 and 200 copies at week 24. So whether or not those 2 individuals are actually going to fail their therapy is not clear. Of more concern were the 3 people that he called virologic failure. One of them had the higher viral load category and 2 of them had lower viral load categories. They also had more than 200 copies of their virus at week 24, but the concerns were patient number 4 and participant number 5 had some mutations on that virus that may compromise the regimen. So that gives us a little bit of pause as to whether or not this strategy is going to be successful. And we always urge that things are not ready for prime time until they're studied in a very large trial so that we can feel confident about the results. And so those studies are ongoing. There are 2 trials called the Gemini Trials that are looking at that drug called dolutegravir, and then again, either together with 3TC alone or together with tenofovir and emtricitabine. Those studies have been fully enrolled and have met their primary endpoint and are going to be presented in Amsterdam this year, so we'll be able to feed back that information to you. So the other question is, well, maybe it's not smart to start 3 drugs at the beginning, or I'm sorry, start 2 drugs at the beginning, but let's go with tradition. Let's people on 3 drugs, get the virus under control, and then drop one. And can you maintain them? And we call that kind of an induction and maintenance kind of therapy. And so again, people tried to do this with many of the different combinations over the years. And again, in the early days, they were not all that successful. But more recently, and I show you here, 4 different studies using different protease inhibitors together with 3TC or 2 nucleosides. These are reasonably large trials, and this is something that we call a plot study or a meta-analysis test. And basically, the diamond is the end of the results, and the diamond is sort of pooling the studies together, and it is in favor of the 2-drug regimen after your viral load has been suppressed. So there is another strategy that might be considered. Don't start 2 drugs, but get people under control and then drop 1 drug. And again, these studies are in their development. We need to learn more. We need to follow these studies over the long term, but it is some information that is supporting that we don't always need 3 drugs. And then the largest clinical trial that's been done to evaluate this approach is something called the SWORD studies. These were 2 identical studies that occurred in different parts of the world. And basically, it took people on triple therapy who had been suppressed on their combination. They either continued that combination or they switched to dolutegravir, the integrase inhibitor I talked about, together with an NNRTI called rilpivirine, so it was a 2-drug combination. So the randomized portion of the trial compared those 2 arms, and then at week 148, everybody who continued to do well was offered the opportunity to go on to the 2-drug combination. And the bottom line, and again, these graphs are something we all need to get used to, but if you look at the bars on the virologic outcomes, none of us need to be a statistics person to show that those 2 treatment arms are performing the same. The other way we express that in statistical means is by the type of graph on the right, where we have a point estimate, what was the real difference between the 2 treatment arms, and then something that we call a confidence interval that tells us how confident we are in the results. And if those bars cross the 0 point, then by definition, the 2 treatment arms are non-inferior. So one treatment arm is no worse than the other, and that's what was found in the SWORD studies. So if you're going to drop to 2 drugs, other than saving costs and saving potential drugs for the future, we would like to also see some benefit in terms of adverse events. And the only one that has been looked at so far, next slide. The only one that's been looked at so far in the SWORD study is that of kidney and bone. So remember that tenofovir is often a drug that's included in the patient's combination, and the tenofovir drug has been linked to differences in bone mineral density and differences in leakiness from the kidney. And this study, a subset of this study, demonstrated that by dropping the nucleosides and just continuing on 2 drugs, that there were some minor improvements in markers of bone health and kidney health. And again, the caution is that these studies are short-term. These are markers of bone and kidney health, and whether or not that will translate into clinical differences in the big picture is still a question open to debate. So, 2-drug therapy, I would say that this is an emerging paradigm. I think I've been able to outline to you that it's really not the number of drugs, but rather the potency of the drugs and the forgiveness of the regimen, as well as the barrier to resistance, which is important. For whatever reason, in all of the studies that we've done with 2-drug therapy, having a member of the reverse transcriptase inhibitor class seems to be important for these things to work. The strategy seems to work in those with viral suppression, but the data for those who have not yet started antiretroviral therapy is still emerging, and we wait for those results in Amsterdam. I thank you.
I'm back. Hi. Thank you very, very much, my esteemed colleagues. I feel like that's a science class in between a little theater arts. So just to bring the session to a close before we go to questions and answers, I did want to just very briefly share the second thought I had in reflecting for today. So although we know that there's still a lot of work left to do to ensure that all people living with HIV can experience the benefits of the evolution of treatment that we're talking about today, the truth is that people are diagnosed now against the backdrop of U equals U, can't pass it on, and Conchita worst. Yeah, somebody knows what I'm talking about. Okay, and in the same way that the experiences of those who came 10, 20, 30 years before me are quite different than my own, the same can be said for those who come 10, 20, 30 years after. There will be a day in the decades to come when the people living with HIV in this room are all the beneficiaries of the fight for effective treatment, as opposed to those who participated in the fight itself. And so standing here in their debt to all those who fought the fight, who shaped all of our lives here in one way or another, I think that it's fair to pause and ask a couple questions. What is it that they have given us, all of us? In some cases, they've helped give us life itself. And what is it that we might lose if we are not good stewards of their legacy? The slide above me contains a photo of a work produced by renowned Cuban-American HIV-positive artist, Felix Gonzalez Torres. The work is entitled, Untitled, Portrait of Ross in LA, and is a representation of Felix's partner, Ross Laycock, who died of AIDS-related illness in 1991. The installation is comprised of candy wrapped in multicolored cellophane and weighs 175 pounds, representing Ross's ideal weight before he died. Viewers of the installation are encouraged to take a piece of candy with a diminishing weight representing Ross's weight loss and suffering in the days leading up to his death. However, each night, the artist stipulated that museum staff replenish the pile, in essence, giving Ross perpetual life. With each step forward, with each ounce of progress that we make collectively together, we all, in a sense, are reaching into that deep, beautiful, messy pile of life, grabbing a sweet, colorful candy and consuming it. But we're not just regular gallery goers that pop in and out. And nor are we just scientists, healthcare providers, service providers, people living with HIV, or activists. We are co-curators of a movement, its history, and its evolution. And with that comes responsibility. A responsibility to ensure that as the pile empties throughout the day, that we all replenish it together each night, so that those who come tomorrow and the day after and the day after that know that we were here. Thank you. So, I want to thank all of my esteemed panelists, Dr. Jean-Pierre Routy, Dr. Jonathan Angel, and Dr. Sharon Walmsley. We do have time for questions from the audience, and so if you have a question from the audience, actually I don't really know what you're supposed to do if you have a question from the audience, but we will make sure you get to ask that question. If you have a question, maybe just stand. Oh, this is rough. No. Oh, do you have a question, Sarah? Okay, great. Do we have a mic? Oh, I think a mic is coming to you.
Thank you. My name is Sarah. I work at YouthCo. We're a youth-led agency working with youth impacted by HIV and Hep C. One of the big questions I get from youth, aside from when will we have a cure, is how can they reduce the number of pills they're taking each day? And so I've been looking at some of the recent research around desimplifying HIV meds, and I'm wondering if the panel can talk a little bit about that work and how it relates to the evolution of HIV treatment.
So there's a couple of issues. One is reducing the number of tablets, and the other is reducing the number of drugs. So over the years, there's been a big attempt to decrease the number of tablets and to decrease the number of times people have to take pills. So that's really been an advance that's been going on over the last decade or so. The downside of that is they get bigger, and some people complain it's like swallowing a pillow every time they have to take their medication. The work I alluded to is an attempt to decrease the number of drugs. So that's where the research is now. We have not been successful with that in the past, but with some of our newer agents, is it now possible for us to decrease the number of drugs? I think perhaps the other more exciting ways in which HIV treatment is evolving is looking towards more long-acting agents. So drugs, either pills or injectables that may last for days or weeks or months. Also different delivery systems, so perhaps reservoirs that might go under the skin like birth control in order to slowly release drugs over a period of time. And I think that those types of delivery systems will be very exciting for all of us, not only for treatment, but also for prevention.
Just to add very little, I think the question is going to be very specific to the individual and why they're on multiple pills, because virtually everyone that's starting therapy for the first time should be able to start a single tablet regimen. Now, as Sharon alluded to, the size might preclude that, but it is an option. So it really goes to what the individual wants: a single large pill or two smaller pills, and different people want different things. But if it's fewer in number of pills, virtually everybody, particularly that are just starting therapy, will have should have a number of options available to them.
Okay, thank you very much for that answer. Is there somebody else who would like to ask a question? Oh, I think right here. Do you want to put your hand up so the runner can see you? We've got somebody just up to the front center.
You know you get a prize if you answer a question. Ask a question.
Answer a question.
Hi, I'm Ray. I work at the AIDS Committee of Durham Region as their youth transitioning coordinator for all of Ontario. My question is that a lot of my youth are asking, they heard about the injection drug that lasts a month or 3, depending on which thing they're reading. What's the news on it? Just curious.
So I think we have good news. It's feasible to have an injection drug, at least 2, mixed in one of right and left. And the LATE-2 study and the new phase of the study is very encouraging, and at least people who are on the studies that I know, so my patients, are very pleased, and I call this type of treatment hands-free. It's like for a phone, you don't need your hand to be treated. And also psychologically, you put all your concern in the hospital or in a clinical center. When you go home, when you go to your private life, you have no pills that can identify you as having a virus that need to be treated. And I think despite being intramuscular injection, the pleasure, at least for those who want to participate, to be on this trial is more than I was expected. And I think that's a big new challenge, and drug companies are looking at that, and will be so important for adolescents when they want to stop medication, for some people with psychological issue, and there is that will increase 10% that we discussed this morning that we cannot reach or treat accordingly. And I think that's a big of hope to improve the 90-90 or 100-100-100.
So I'll answer it from a bit of a different perspective. In case the question is, when will it be available? And it's difficult to give a time. Certainly, my view is the same as Jean-Pierre with the positive responses. People have those that are interested in injection. It's not for everybody, but it's got to be people that are willing to be injected or prefer to receive an injection. You referred to the LATE-2 study, which was a phase 2 study that showed that it was at least as good as oral therapy. There are 2 ongoing registrational studies, so those are the studies that are large enough to get submitted to the regulators for approval. Those are ongoing, and and so far the information looks good from those, but I can't tell you that it's going to be licensed. I think people are predicting that it will be licensed. The timeline, again, I can't tell you. Someone from ViiV may be able to give you better timelines. I would say at least a year before it's licensed. That's a guess. It will be licensed first every month. It'll eventually, probably, if things continue to go well, be licensed to be given every 4 weeks. Probably be licensed to be given every 8 weeks. Again, it's all speculative, right? If everything continues to go well, there will be a lot of things. I mean, licensure is the first step, and there'll be a lot of other things that need to be sorted out. So reimbursement's always an issue for drugs. But then who's going to give the injection? Right now, in the setting of clinical trials, it's being given by someone, a nurse or a physician or another healthcare provider. It tends to be larger centers in the context of a clinical trial. How that will be rolled out into the community, remains to be seen. And there's going to be a lot of discussions between ViiV, whose compound it is, physicians, nurses, pharmacists, infusion centers, to try to sort. And so that's something that has not been sorted out.
Do you think it would ever become, because for the injection-wise, as you were saying, that you don't know who will be providing it, would it be possible to do it like an insulin in a
No.
sense? No?
No, so that's larger they're larger volumes, and it's intramuscular. So the current drug that'll be like the current, yeah. So that will not be it's not feasible to self-inject, yeah.
But that's not to say that drug development may not be there at some point, but the ones currently under investigation are intramuscular, so they're not amenable to that.
Sherry? Yeah.
Big voice.
Yeah, I can read your mind.
But use it anyway.
And your lips.
I might have missed it in the talk. First of all, my observation is that these questions are coming from young people. And I have a close connection to young people who are positive also, many of whom are heavily treatment experienced. And I'm just wondering, I might have missed it in the talk, but what the indication for the long acting is going to be. Is this only going to be for naive, or are we looking at potentially using it for heavily treatment experienced patients like youth who have serious adherence issues?
So the current evaluations of the compound are in either naive patients who have not been on drug or switch for people who are suppressed. That's not to say that the drug would not be an option in terms of what we call salvage is a bad word, but extra therapy, but it will need other partners. So it will need other drugs, and that would obviously be dictated by the individual case and what resistance they have. So it certainly could be a component of that kind of therapy, but will be needed to be partnered with other compounds.
So, Sherry, the label, we don't know what it'll say for the label indication, but that will likely reflect very closely what the clinical trials are. And for the naive, it's straightforward, but for the switch studies, the studies are quite restrictive. The people enrolled in the clinical trials have to be either on their very first or their second antiretroviral regimen. So not a lot of drug exposure for the clinical trials. The label may be restrictive to be that. I can't, I can try to predict, but I don't know. What then will likely happen once it gets into clinical use, like a lot of other medications, they're used kind of, quotes, off-label. And I think with real-life experience, we'll learn who it should or should not be used in and how it should be combined with other medications. But I think initially the label will be quite restrictive.
Okay, great. Any other questions? So far, the middle has been representing. So I don't know what's happening on the sides of the room. Anyone? Am I missing a hand? Oh, maybe not. I don't know. Hi. I just met these lovely people today. Listening to one another's presentations, was there anything that kind of popped up that you would like to ask each other about, or?
I think I'd just like to continue to push. I mean, lots of people say there's no more room for research in HIV, and I think that this just demonstrates there's lots of room for further research in HIV, and we continue to have new products to look at. We continue to look at new ways in which we may combine them, or new ways in which to manage them or deliver them. So even though our drugs are doing great with 99% response rates in many of the clinical trials, there's still more that we can learn, and there's still more that we can do to optimize therapy in the long term.
So one comment on a lighter regimen or new regimen is to be sure that we block the virus in non-body parts. So lymph node, colon, we need also not just to have a blood test, but to be sure that we may maybe with new compound impact the reservoir, and be sure that in some tissue we have the appropriate drug distribution than in a blood, and there is still some debate. Of course, it's very difficult to do the sampling for participants, but that also a word of caution to put a light regimen. They have to be proven and have also time lag. We need 2-3 years to be sure that less can be as good as more.
And I always say I want to add an extra 90 to 90, 90, 90. It's 90 alive at 90 and being well.
Well, hats off to that. I'll take that, yeah. So we've got about 5 minutes, and so enough time perhaps for one more question, if anything's arisen. I don't believe so. So, do you have anything else that you'd like to say?
I think we should close.
Okay, well then we'll give you the extra 4 minutes of your time back. You've been a lovely audience. Thank you so much, ViiV, for sponsoring this symposium, and enjoy the rest of CAHR.